Earlier this year, there's a scientist called Kevin McKernan.
I have 25 years of experience in the genomic space. I've worked as a team leader of R&D at the Human Genome Project at Whitehead and MIT, and I have over 57,000 citations to publications in my space and multiple patents on PCR and sequencing.
He was running an experiment and he needed a control, and he just yanked a couple of vials of the mRNAs, the Pfizer and Moderna. He discovered DNA fragmentation in these vials that's not supposed to be there.
And to my shock, discovered that the expression vectors for the vaccines are still in the vials.
So essentially they're contaminated. And sure enough, around the world over the months, bit by bit, people started replicating his findings.
There's lots of studies now coming out about this floating DNA.
Wir sind in einem Privatlabor in Magdeburg. Professorin Brigitte König untersucht hier Corona-Impfstoffe. Das Ergebnis: alle Proben sind verunreinigt mit Fremd-DNA.
In every laboratory in the world that's tested the vials so far, they're all contaminated with DNA.
My name is Philip Buchholz. I have a PhD in biochemistry and molecular biology.
And he just testified in the South Carolina Senate to say that he's confirmed Kevin McKernan's findings.
The people in my laboratory were really good at detecting foreign pieces of DNA in places where they're not supposed to be.
We know that Pfizer, when they ran their randomized control trial, they used one particular process for manufacturing the mRNA that goes into the vaccine in the end, and it's a very clean process.
There was no plasmid DNA to contaminate the stuff that was used for the trial. But that making that PCR product doesn't scale the way that was necessary to vaccinate the whole world.
Now, out of nearly 44,000 participants, only 252 participants received the vaccine that was manufactured by what's called the scale-up process.
So a cheaper way to scale up the production of this template is to clone that PCR product into this plasmid vector, put the plasmid vector into bacteria, and then you can grow up big vats of the bacteria. They make a lot of the plasmid DNA for you. Then you use that plasmid DNA as the template to drive this transcription reaction.
What it looks like is that the DNA cleaning process wasn't satisfactory.
Now we're seeing that instead of it getting cleaned out as we were told the way the product's supposed to be made, we have all this residual DNA.
The pieces are very small because during the process they chopped them up to try to make them go away, but they actually increased the hazard of genome modification in the process. And that's how the contamination ended up in the production batches, even though it was not in the stuff that was used for the authorization trials.
The current regulatory limits for DNA fragmentation or residual DNA in vaccines is for naked DNA, not DNA wrapped in lipid nanoparticles.
Naked DNA gets chewed up immediately upon vaccination, and there's no real mechanism for it to get inside the cells. They inappropriately applied that regulatory limit to this new kind of vaccine where everything is encapsulated in this lipid nanoparticle. It's basically packaged in a synthetic virus able to dump its contents into a cell.
And it looks as though the regulators have not accounted for a lipid nanoparticle casing around the contamination.
We do this in the lab all the time. We take pieces of DNA, we mix them up with a lipid complex like the Pfizer vaccine is in. We pour it onto cells, and a lot of it gets into the cells, and a lot of it gets into the DNA of those cells, and it becomes a permanent fixture of the cell. It's not just a temporary thing, it is in that cell and all of its progeny from now on forevermore. Amen.
I mean, it's even beyond reckless what we're seeing. This is the risk of moving at the speed of science.
Right.
That you're playing willy-nilly and loose with this risk of human DNA integration.
But it's pretty reasonable based on solid molecular biology that it could cause a sustained autoimmune attack toward that tissue. It's also a very real theoretical risk of future cancer in some people. It could be causing some of the rare but serious side effects like death from cardiac arrest. So there's a lot of cases now of people having suspicious death after vaccine.
What we see a lot with the bureaucratic response is there's no evidence of that. But the question is, well, have you tested for it? And I think that the answer is no.
Somebody should go about sequencing DNA samples from stem cells of people who are vaccinated and find out if this theoretical risk has happened